Start. All right, so a distinct Fusobacterium nucleatum clade dominates the colorectal cancer niche by Eric Kraus. So good morning, and this is my project. What were the researchers trying to determine? Well, that would be the role of Fusobacterium nucleatum, what role it plays in the development of colorectal cancer. Now, why should we care about this? Because in 2025, the American Cancer Society projects there will be approximately 154,000 new cases of CRC and 53,000 deaths from this disease in the US alone. CRC remains a significant health concern, ranking as the third most common cancer and third leading cause of cancer death in both men and women. So, along with 154,000 new cases, subsets of those 107,000 colorectal cancer, colon cancer rather, and then 47,000 rectal cancers. We're going to see 53,000 deaths out of that. 1 in 24 men and 1 in 26 women are going to develop this. Increased incidence in younger adults is the big issue I picked this because there's a concerning trend of increased colorectal cancer rates in younger individuals, particularly those under 50. Screening rates, even though they've improved, there's still a significant number of people over 45 who are not being screened as recommended. Key findings: F nucleatum levels in CRC tumors were linked to recurrence, metastasis, and worse patient outcomes. Pangenomic analysis of 483 genetic factors that are enriched in CRC-associated F nucleatum strains. F nucleatum subspecies animalis dominates in CRC tumors, but is split into two clades, being clade 1 and clade 2, with clade 2 being the only one that's prevalent in observed tumors. Clade 2 has 195 associated genetic factors that enhance its metabolic potential and ability to colonize the GI tract. Experiments showed clade 2 treated mice developed more intestinal adenomas and had altered metabolites. Human CRC tumor analysis of 116 patients and stool samples from 627 CRC patients versus 619 healthy confirmed clade 2 enrichment in tumors compared to normal tissue. This study highlights very well clade 2's specific role in CRC and its genetic adaptations to the tumor environment. What is Fusobacterium nucleatum? Is an oral bacterium that has been increasingly linked to CRC. Gram-negative anaerobic bacterium commensal to the human oral cavity. Plays a prominent role in periodontal disease and key component of periodontal plaque. Creates a pro-inflammatory environment conducive to tumor growth and is a major contributor to mouth cancer, as you see on the right-hand side in the middle picture. Why is it such a little problem? Well, it shows it is more abundant CRC tumors compared to healthy tissues. High levels in CRC, bad news. Persistent metastasis disease targeting this particular bacterium through microbiome modulation may help, maybe diet. Strain variations in its genetic and phenotypic traits create challenges in consistently reproducing the effects. By using a large collection of CRC-derived FN strains and comparative genomics, the study authors identified a specific and highly virulent clade that dominates in CRC tumors. This clade is recognized as the focus of future research in F nucleatum's role in colorectal cancer and for the development of targeted treatments. All right, so using classical microbiome to culture and analyze F nucleatum strains. Here, the researchers use conventional microbiology culture methods to obtain the necessary bacteria of interest to facilitate proper execution experiment. Out of 135 tumors, 65 strains were isolated from 59 patients. 81 were obtained from repositories to use as controls. Back biome sequencing we used to generate a complete methylomes for 146 unique strains. 92% of those were F nucleatum. The analysis showed CRC strains had smaller accessory genomes than oral, with 483 gene clusters in CRC versus 241 in oral. F nucleatum subspecies animalis was found to be significantly enriched in CRC tumors, and animalis had the smallest core genome, indicating genetic heterogeneity. So strains like FNA 7_1 were used for the study. The analysis of virulence factors had no significant enrichment in FNA strains compared to other subspecies, indicating other genetic factors drove the association with CRC. All right, so figure 1A, we got a schematic of Fusobacterium strain collection of 146 samples and a sequencing strategy for unique strains. Single molecule real-time sequencing was used. Over at B, we got a column graph depicting the proportion of Fusobacterium genomes, subset by species, with CRC being in orange and oral being in blue. C describes the composition of FM pangenome subset by niche, Amvio gene clusters, and GC prevalence was used to define core, greater than 95% accessory, greater than 5%, less than 95%, and rare, which is less than 5% features conserved in both CRC-associated and oral-associated strains. D gave us the proportion of niche-enriched gene clusters across CRC-associated versus oral-associated genomes. Plot box shows 25th percentile median and 75th percentile. In E, we got the COFAM, Koala, KEGG, ortholog analysis 27 niche-enriched gene clusters. F gives us a column graph depicting the proportion of FN genomes grouped by subspecies within the CRC cancer and oral niches. G is going to give us the gene presence versus the absence heat map of canonical FN virus factors. The heat map is organized using RPOB gene-based phylogenetic tree. And for each tree, the tree endpoints of the niche origin. CRC is orange, oral is in blue. All right, FN nucleatum clade 2 enriched in CRC niche. The researchers investigated the genetic and ecological distinctions between two clades of F. nucleatum, subspecies Animalis, clade 1 and clade 2 in CRC. They found analysis of housekeeping genes confirmed two distinct clades, C1 and C2. The average nucleotide identity between the two clades was 91 and 93%, indicating significant genetic divergence. The two clades have distinct protein coding gene profiles indicated by gig map and component analysis, with VNA 7_1 being grouped in clade 2. C2 is an enhanced adaptability for CRC colonization due to larger accessory genome, chromosome size, and more plasmids and genetic defense elements than C1. Epigenetic differences were revealed with specific DNA methylation motifs driving the distinction between the two. Even though both clades are present in oral cavity, only C2 is significantly enriched in CRC tumors. This was supported by 16S RNA sequencing, and clade 2 was shown in tumor samples but not in saliva samples. We'll go through this a little bit faster for figure 2. K-SNAP 45 maximum likelihood in A, whole genome phylogenetic tree. For each genome, which was 135 samples, tree endpoints indicate the niche, which was CRC is in orange and oral was in blue, and the bar color indicates the FN subspecies. The background color indicates the FNA clade. FNA clade 1 is in green. Sorry about the skip. And clade 2 is in lavender. B, cluster dendrogram with ANI matrix. The clades are highlighted with green and lavender boxes. C is going to give a gig map visualization of the protein coding gene content across FN genomes. Dot bar color indicates subspecies. We're concerned with clade 1 and clade 2, and the box indicates, once again, clade 1 is green, clade 2 is lavender. So make a mental note of that, because that's what I'm going to say from here going forward. PCA of ambio gene clusters by presence in D and absence in each genome. Colors indicate FN subspecies and clades. Ellipses are drawn to 95% confidence. On E, at the left, we have PCA of FNA genome-wide methyl-modified mononucleotide sequences. Ellipses are drawn to 95% confidence, and the overlay the PCA biplot shows top 5 nucleotide motifs that are driving FNA clade bifurcation. At the right, a table indicating the distribution of each clade motif across each clade. F gives us a column depicting the proportion of FNA CRC associated and CRC associated and oral associated genomes subset by clade. So FNA enrichment where it's not supposed to be. Here the explorers explored why Fusobacterium nucleatum subspecies Animalis clade 2 is enriched in CRC tumors compared to clade 1. Key findings were mice treated with the mixtures of clade 2 strains exhibited more intestinal adenomas in the large intestine versus clade 1 treated mice or the control group. Clade 1 showed no difference from the control. Metabolomic analysis of roughly 1,300 metabolites inferred distinct metabolic profiles in the C2 treated mice, which clustered separately from the C1 treated and control. Clade 2 treated mice exhibited enriched glutathione metabolism and gamma glutamyl amino acid pathways with increased precursors, cysteine and gamma glutamylcysteine, reduced glutathione, higher levels of degradation products. The reduced and non-reduced glutathione ratio increased 3.5 to 3.0 fold compared to control group C1 and C1 groups. So in NOVA, the p-values were 0.0031 and 0.0047 indicated elevated oxidative stress. Clade 2 mice treated mice showed increased markers of oxidative stress, decreased polyamines, putrescine and spermidine, which combat oxidative stress and resulting inflammation. The treated mice also exhibited higher levels of pro-inflammatory metabolites, which promote oncogenesis metastasis. These metabolic changes suggest clade 2 creates a pro-oncogenic intestinal environment by enhancing oxidative stress, inflammation, pre-tumor progression, unlike clade 1, which lacks those effects. So now we're going to look at figure 3. A is going to give you a gene presence versus absence column graph depicting the proportion of the genomes containing canonical FN virulence factors subset by clade. At B, we got left computational confocal analysis of colon cancer epithelial cells on co-incubated with representatives clade 1 or clade 2. Right, a bar plot demonstrating the percentage of HCT116 cells with intracellular FNA with sample size of 3. In C, a p-pangolin map of the FNA genome. Each node represents a gene group, and synthetic nodes represents neighboring genes. The size indicates relative presence across the FNA genomes, and the color depicts pangenome partition, with FNA core, which is in red. Clade 1 ancestry genomes in green, clade 2 is in lavender. White arrows and oblongs in the right panel indicate FNA clade 2 associated putative EUT, and PDU operons. D gives you schematics of the operons, clade 2 operons, and ENF differentially express genes in a representative clade 2 strain, SB010 exposed to E8 and E or 12PD, and F compared to unexposed SB010 control. How clade 2 affects tumoral tumorigenesis. Here, the investigators, researchers investigated the impact of FNA nucleatum subspecies clade 2 versus clade 1 intestinal tumorigenesis and metabolism in a mouse model of CRC. They found that in a dextrin sodium sulfate-induced colitis mouse model, mice treated with a mix of 3 clade 2 strains showed significantly more intestinal adenomas, particularly in the large intestine, compared to clade 1 treated or control mice. Clade 1 showed no significant difference from controls. Metabolomic analysis of 1,300 intestinal metabolites revealed distinct metabolic profiles in clade 2 treated mice clustering separately from clade 1 in control. Clade 2 treated mice exhibited enriched glutathione metabolism and gamma glutamyl amino acid pathways. With the reduced and non-reduced glutathione rates, ratio increased 3.5 and 3.0 fold compared to control, and the clade 1 groups indicate elevated oxidative stress. Clade 2 treated mice also showed increased markers of oxidative stress. For example, cysteine and cysteine glutathione disulfide, and decreased polyamines such as putrescine and spermidine, which combat oxidative stress and inflammation. Leaving us with higher levels of pro-inflammatory metabolites, so N-monomethylarginine, dimethylarginine, prostaglandins, ceramides, and eicosanoids, which promote cancer cell proliferating metastasis. These metabolic changes suggest clade 2 creates a pro-oncogenic intestinal environment by enhancing elevated oxidative stress, inflammation, tumor progression, unlike clade 1, which lacks these effects. So figure 4 gives us a schematic of the study of 6 to 8 week old mice receiving streptomycin and dextrin sodium sulfate treatment to alter the native microbiome and induce colitis. The mice were orally gavaged with vehicle control arm 1 and mix of 3 represented clade 1 in arm 2 and clade 2 in arm 3 strains. A strain mix was used to capture a higher proportion of FNA clade-specific accessory genes. The mice were monitored until the endpoint 6 weeks post-gavage, and they reached 15 to 17 weeks of age. Over in B, it gives us a plot indicating the number of adenomas in the large intestine by treatment arm. Vehicle controls gray, and clade 1 is green, clade 2 lavender. 8 mice per arm. C gives us partial least squares dominance analysis of detected intestinal metabolites, 1,300 samples. The colors represent the treatment arm, as I've just said, gray, green, and lavender. Clade 2 enrichment in human CRC cohorts. Here, the researchers examined the prevalence of Fusobacterium nucleatum subspecies clades 1 and clade 2 in CRC tissues and stool samples, building on findings that clade 2 is enriched in CRC and promotes tumorigenesis in mice. Researchers found that 16S RNA sequencing of tumor tissue from 116 treatment-naive CRC patients and paired normal tissue from 62 patients showed F nucleatum was significantly enriched in tumors with a p-value of 0.0022. Using FNA clade-specific amplicon sequence variants, only clade 2 was significantly enriched in tumors compared to normal tissue with a p-value of 0.0093, unlike clade 1 and other subspecies. Stool metagenomic analysis from 627 CRC patients versus 620 healthy individuals showed FNA in 29.2% of CRC stools versus 4.8% of healthy stools. Clade 1 and clade 2 were associated with CRC, but clade 2 had a larger effect size, 0.45 versus 0.21. When clade 2 was absent, clade 1 showed no significant CRC association. Clade 2 was more prevalent in abundant CRC patient stool samples than clade 1. clade 2 detected independently in 147 samples, co-occurring with clade 1 in 31 and clade 1 alone only in 5. These findings confirm clade 2 as the dominant CRC-associated clade, supporting its higher virulence and tumorigenic potential observed in vitro and in vivo studies. Figure 5 gives us plots in A showing the relative abundance of Fusobacterium species with FN subspecies clades in the right plot using microbial 16S RNA gene sequencing of paired tumor, which is in orange, and normal adjacent, which is black tissue. We had 62 patients with CRC. Amplicon sequence variants were used to obtain clade resolution. B, you have the plot showing the relative abundance of clade 1 and clade 2 within patient primary colorectal cancer tissue from independent cohorts. In C, we got clade 1 and clade 2 detection in stooled metagenomic data from patients with CRC in healthy individuals. Left plot shows the pooled effect size of clade 1 and clade 2, calculated using a meta-analysis of standardized mean differences, species levels, genome bin difference abundances in all CRC samples, which 627 of the CRCs, and healthy samples 619. Right plot shows the effect of clade 1 and clade 2 calculated using the same approach, but here samples in clade 1 co-occurred with clade 2 were excluded. The statistical significance was assessed by the Wald test, two-sided. All p-values corrected using the Benjamini-Yucatelli method. Over in D, we have clade 1 and clade 2 in presence of stools and metagenomes of patients with CRC. The bars indicate individual stool samples from patients, which is 627. Lower brackets indicate the number of stool samples in which C1 occurred independently in 5 samples, clade 2 independently, 147 samples, and clades co-occurred in 31, or clades were not detected, which was 444 samples. All right, so the meat of this paper, the methods, what do they do and how do they do it? So Fusobacterium strain isolation from tumor tissue from patients with CRC. The process of isolating Fusobacterium strains from colorectal cancer tumor tissue from parents in North America and Europe. Tumor tissue was minced and spread on selective fastidious anaerobic agar, which is FAA, plates supplemented with 7 or 10% defibrinated horse blood, which we're going to abbreviate as DHB, and antibiotics, which were josomycin, vancomycin, and norfloxacin. Plates were incubated anaerobically at 37 degrees Celsius, checked for growth every 2 days, and they were streak purified. Colony PCR with 16S rRNA gene primers, which were primers 342F and 1492R were used, followed by Sanger sequencing and blast-in analysis to confirm bacterial species. Cultures were stored in tryptic soy broth at 40% glycerol, negative 80 degrees Celsius. Fusobacterium strain isolation from Korean collections. So here they collected samples from the Fusobacterium strains from the Korean collection of for oral microbiology, KCOM, and American type culture collection, ATCC. KCOM strains were isolated from the oral cavity as previously reported. Strains from both repositories were grown on Shadler agar plates supplemented with vitamin K1 and 5% defibrinated sheep's blood on a fastidious anaerobic agar plates with 7% defibrinated horse blood. They were incubated at 37 degrees Celsius for 5 to 7 days in a Bactron 600 anaerobic chamber, then stored in Shadler broth with vitamin K1 at 30% glycerol at negative 80 degrees Celsius. Here they had high molecular weight genomic DNA extraction. Here the section details the culturing and DNA extraction for Fusobacterium strains. Strains were grown anaerobically at 37 degrees Celsius for 48, 72 hours on FAA plates, supplemented with 10% DHB. CRC-associated strains were grown on plates with additional antibiotics, josomycin, vancomycin, and norfloxacin at 3, 4, and 1 microliters, respectively. High molecular weight genomic DNA was extracted using the Master Pure gram-positive DNA purification kit, with cells from 2 plates resuspended at 1.5 milliliters of PBS centrifuged and processed with double reagent volumes and no vortexing to avoid DNA shearing. DNA was quantified using a qubit fluorometer. The PacBio single molecule real-time sequencing and genome section describes the sequencing of Fusobacterium strains using SMRT sequencing on a PacBio Sequel or SSequel2 instrument at the University of Minnesota Genomics Center. Sequencing reads were processed with the microbial assembly pipeline and Pacific Biosciences smart analysis pipeline. Here we have Fusobacterium species typings in slide 18, okay? This section outlines the subtyping Fusobacterium genomes to species, F nucleatum genomes to subspecies level using cumulative score from 3 marker genes, 16 sRNA, RPOB, and zinc metalloprotease gene. Confidence score for each species or subspecies was calculated as the number of concordant genes results divided by the number of marker genes present. Results are listed in supplementary table A in the paper if you want to reference it. Pangenome analysis was using ANVIO, pPangolin, and GigMap. Analysis of the pangenome across 135 genomes and the FNA pan genome across 51 genomes. For FN, ANVIO used a, you guys can see this. I'll let you guys go through that any skip time. So in a genomic dendrograms, this section describes the phylogenetic analysis of 135 Fusobacterium nucleatum genomes. Individual genes and gene sequences were aligned, protein sequences were aligned, and a maximum likelihood dendrogram was created. Additionally, KSNAP3 with the K-commerce size of 13 was used to generate maximum likelihood phylogeny for the genomes. Identification of FN canonical virulence factors. Here we have the analysis of canonical Fusobacterium nucleatum virulence genes in the collection of the FN genomes. The operon contextualization across prokaryotes to uncover synteny tool was used, octopus, was used of the maximum percentage threshold identity threshold of 60% to identify the presence of these virulence genes. The identification of the FN genetic defense systems and prophage section have the analysis of bacterial defense systems and prophages in Fusobacterium genomes. The prokaryotic antiviral defense locator tool was used to identify innate bacterial defense systems with the phage search tool. Enhanced release was used to detect intact prophages. In PCA, this section describes the principal component analysis performed of Fusobacterium nucleatum and its subspecies Animalis. In the FN and HCT 116 co-culture assays, the HCT 116 cells were cultured on McCoy's 5A medium agar with 10% fetal bovine serum at 37 degrees Celsius and 5% carbon dioxide seated at 1.25 times 10 to the 6 cells per well. 6 well plates with glass cover slips allowed them to adhere for 16 hours. After co-incubation, the cells were washed, fixed with 4% paraformaldehyde, and stained with nuc blue and actin green for DNA and actin visualization. Here we computational analysis to determine intracellular FN, and this section describes the analysis of confocal Z-stacks from co-cultures of HCT 116 colon cancer cells with Fusobacterium nucleatum subspecies Animalis clades 1 and 2. Bacterial surface volumes were created using FM 4-64 FX membrane stain fluorescence, and eukaryotic cells were identified using nuclear and actin stains with nuclei split by seed points. The percentage of HCT 116 cells containing intracellular FNA bacteria was quantified and compared between clades 1 and 2. The cell length and width measurements. This section describes the measurement of cell dimensions of Fusobacterium nucleatum subspecies animated clades 1 and 2 using Fiji with the bioformats plug in to import Alicia files. Large scale was set to 1 millimeter equals 9.979 pixels. Cell lengths and widths were measured using the freehand straight line tool, targeting the brightest points on the cell membrane stain. Statistical comparison of cell dimensions between clade 1 and clade 2 was performed using Welch's t-test in GraphPad Prism version 7.0. Here we have RNA sequencing, and it discusses the culturing of RNA extraction of Fusobacterium nucleatum strains SB010 and KCOM3764 for transcriptomic analysis. The strains were grown on FA plates with 10% DHB at 37 degrees Celsius for 2 days. Lawns were prepared and cells were resuspended in tryptic soy broth. The cultures were then split into triplicates and incubated anaerobically at 37 degrees Celsius for 4 hours in TSB alone. TSB was 50 millimoles of 1,2-propanediol and 20 nanomoles of vitamin B12, or TSB with 15 millimoles of ethanolamine and 20 nanomoles of vitamin B12. Cells were pelleted, washed in PBS, and stored at negative 80 degrees Celsius. The RNA was extracted using RNeasy extraction kit with ribozero plus RNA depletion and sequenced to a minimum of 12 million paired end reads using Illumina standard RNA library preparation. In the mouse model experiments, the section details an experiment using mice to study the effects of F nucleatum clades 1 and 2 on intestinal tumorigenesis. Female mice that were 6 to 8 weeks old were divided into 3 treatment arms across 2 trials. The mice were then pre-treated with streptomycin, 2 milligrams per milliliter, and 1.5 dextrin sodium sulfate in drinking water for 7 days each to induce colitis and promote tumors, followed by normal water for 24 hours before oral gavage with 200 microliters of the respective treatments. FNA strains were cultured on FAA plates with 10% DHB at 37 degrees Celsius and Bactron 600 anaerobic chamber for 2 to 3 days, resuspended at PBS and standardized by optical density, OD 600 nanometers to ensure equal cell mixes. Mice were monitored for 6 weeks post-gavage till they were 15, 17 weeks old, and the intestinal adenoma alone was blindly assessed in 8 mice per arm. Sorry, I'm just speaking so fast. I got a lot to cover. Intestinal metabolomics analysis profiling was performed on mouse intestinal tissue, 4 samples using UHPLC -MS divided by MS by Metabolon, analyzing over 5,400 metabolites from 70 pathways of both eukaryotic and bacterial origin. The mouse fecal DNA extraction quantity of PCR, we had the DNA was extracted using Zymo-Quick DNA microprep kit, and the Fusobacterium genus DNA was quantified using a custom TACMAN qPCR assay, normalized to genomic DNA, and run in duplicates. Each of the runs included positive and negative controls, as would be expected, and the primer and probe sequences for this assays were specified. Biolog PM10 phenotypic microarray plates, we had the FNA strains were cultured anaerobically on FAA plates with 10% DHB, then resuspended and pre-reduced and normalized. The plates were equilibrated to aerobic conditions for 10 minutes, then incubated anaerobically at 37 degrees Celsius for 24 hours. The absorbance at 590 nanometers was measured to assess metabolic activity. Now, here in the glutaminase assay, the FNA strains were cultured anaerobically on FAA plates with 10% DHB for 2 days, then transferred to TSB with yeast extracted and L-cysteine for 20 hours of liquid culture. Cultures were standardized, centrifuged, and resuspended in a pH of 3.1 GS solution containing glutamine, bromocresol green, sodium chloride, and Triton X100. Each strain was incubated in triplicate in a 96-well plate for 37 degrees Celsius for 2 hours under anaerobic conditions. After centrifugation, the supernatants were transferred to a new plate, and absorbance at 600 nanometers was measured. The acid resistance simulated gastric fluid. Well, they're going to have to do this to see if the bacterium can pass the digestive system, right? So the strains were cultured anaerobically on FAA plus 10% DHB plates for 1 to 2 days, and then transferred to TSB with yeast extracted and L-cysteine for 25 hours of liquid culture. The cultures were standardized at OD 600 equals 1 and exposed to either TSB, simulated gastric fluid at a pH of 3, or gastric fluid with 10 millimoles of glutamate. The patient specimens were all analyzed using tumor samples, diagnosed as colorectal adenocarcinoma. Specimen selection did not consider age, sex, or ethnicity, but treatment-naive primary CRC tumors were prioritized for microbial culturing. Here we have the bacterial 16S RNA gene sequencing, and they obtained data, DNA from patient tissues. It was extracted using and analyzed using Zymo-Biomics, targeting the bacterial 16S RNA V3 to V4 region with custom primers. Library prep included high-fidelity PCR and cleanup to reduce chimera formation, followed by aluminum MiSeq sequencing. The analysis was performed using the NF-Core AmpliSeq pipeline with customized parameters to ensure specificity and quality control. The meta-analysis of FNA clades in relation to CRC using publicly available shotgun metagenomic samples. The researchers describe here in this particular section about metagenomic analysis to assess the association between Fusobacterium nucleatum subspecies Animalis, clades 1 and 2 in colorectal cancer. Shotgun stool metagenomic data from 9 public cohorts, 627 CRC patients versus 619 healthy individuals, was analyzed. Distinct species level genome bins, SGBs, were identified for each FN subspecies. Standardized mean differences and standard errors were calculated to compare CRC and healthy conditions. Mapping of the putative EUT, PDU, and GDAR operons in publicly available metagenomic samples. So we compared the presence of EUT, PDU, and GDAR operons in CRC patients versus healthy individuals, and shotgun stool metagenomic data from 9 public cohorts. Once again, the 627 CRCs versus 619 controls were used and analyzed. Reads were mapped to FNA SB010 operons using bowtie 2, and gene coverage was assessed with CMSeq. Genes were considered detected if coverage breadth exceeded 50%, and the operons were counted as present if over 90% of EUT or PDU genes or 100% GDAR genes match the SB010 operon structure. What a mouthful. Man, they did a lot of work. Okay, so we covered this. So we're going to, they covered, just went over this. Sorry, I didn't click on the slide enough. Click on to the next one. All right, so our discussion. So even though sequencing confirmed to be in tumors, what is the functional role of F-nucleatum clade 2? It serves something, or else it wouldn't be able to get past the digestive system out of the oral. So FN typically found in the oral cavity, but is enriched in CRC tumors. Which Fusobacterium nucleatum strains contribute to CRC? The study identified CRC-enriched genetic factors primarily in a specific clade of F-nucleatum subspecies animalis, as we just covered. The two clades, clade 1, mostly oral, clade 2, dominant CRC tumors. Only clade 2 increased tumorigenesis and oxidative stress in CRC mouse model. Does this directly apply to humans, or is this one of the deals where correlation doesn't equal causation? Clade 2's adaptation to the CRC niche. Specifically, what is it about this particular clade of bacterium that allows it to occupy both the oral and the colorectal areas and become a nuisance in the CRC cohort? Clade 2 is identified as a highly virulent subgroup, recommended as the focus for future CRC mechanistic studies and therapeutic development. I've recently had conversations with Dr. Sabina Zahn over at Progena Biome, who some of you know of her, and her great work within this space. And we are suspecting that it's possible, maybe even likely, that the processed seed oils are somehow contributing to the growth and proliferation of the clade 2 in the human colon. So this is something that has been taken back. So here I came up with 10 thought-provoking questions. I know I only had to come up with 5. I'm not going to cover all of them, but let's look at number 1. How did the 195 clade-specific genes in clade 2 contribute to the selective advantage in acidic colorectal cancer tumor microenvironment, and what insights might this provide into microbial adaptation to other acidic pathological niches? Maybe this thing can be adjusted by diet, less acidic environment in the human. Let's look at 3. Given clade 2's enrichment liver metastasis, what mechanisms might facilitate its dissemination from primary CRC tumors to metastatic sites? And how could this inform strategies to prevent CRC metastasis? What's it eating? What's proliferating this thing? What's causing its oncogenesis? 5, what are the ethical and practical challenges of implementing a qPCR-based diagnostic assay for clade 2 in stool as a routine screening tool for CRC, particularly in low resource settings? 7, what role might diet or lifestyle factors play in promoting the colonization of clade 2 in the gut, and how could these insights be leveraged for CRC prevention strategies? Could we possibly prevent this thing, as I've said ad nauseum, with diet and dietary interventions versus pharmaceutical? We don't know. We got to see. Right? We need funding for this. Number 9, what are the limitations of using mouse models to study clade 2's role in CRC? How might these limitations affect the translation of findings in human patients? Very good question. Maybe we should look at this in pig models as, if possible, as there are a number of pig parts such as heart valves that are usable in humans. Would it have the same response to this particular bacterial clade? And we stay similar. That way we don't have to do so much and impart so much on the human subjects. And finally, 10. How could the discovery of clade 2's dominance in CRC tumors reshape public health approaches to microbiome sequencing, and what broader implications might this have for understanding microbial contributions to other cancers? Very thought-provoking indeed. I would highly recommend, if you're a nutrition nut and biochem nerd like myself, that you look through these references and read some of these papers. They all do, for the most part, have something to do with clade 2 in this particular paper. As I resource a lot of information out of them. There's also a couple links in here to like the American Cancer Society, so you can look at factors that I referenced at the very beginning of the paper. Overall, a fascinating paper, but is very alarming considering our age group. And I would encourage my fellow Gators to do their due diligence. That way, whenever you know of someone, because this will affect someone that you know at some point, and you're able to steer them in the right direction, have a better outcome. I want to thank you for taking the time to listen to my spiel. And I look forward to discussing this paper with you guys in perusal. Thank you. Let's stop the recording here.